All right. So I shared some feedback on your mid-semester submissions. Uh, and one of the reasons, one of the things that I noticed was that actually a couple of things. I mean, some of the submissions were pretty disappointing in the sense that there was hardly any effort. Uh, and you can sense that because it's pretty easy to make out that this was something that was started like a day in advance. And as I've been saying repeatedly, that at least for this course, if you start a day in advance, nothing's going to happen. So those submissions fared pretty miserably as far as grading is concerned. The other thing was that I don't think anybody got what Fisher was trying to say with respect to the nature of evolution on landscapes. Wright's argument, most of you had that right, uh, but actually nobody had Fisher's argument right. Some of you wrote about fundamental theorem, Fisher's fundamental theorem as Fisher's viewpoint, but that's not really it. That wasn't. I mean, fundamental theorem was fine, but that wasn't really the point of contention between Wright and Fisher. Anybody wants to say what Fisher was what the problem of what the contention was between Wright and Fisher with respect to nature of evolution and particularly structure of landscapes? Anybody?
So Fisher had a single peak landscape in mind,
Right.
and Wright, according to him, when epistasis played a role, there will be multiple peaks in the landscape.
Correct. But why does what's important is to really sort of why does Fisher say that it's a single peak structure? I mean, we've seen how epistasis leads to multiple peaks in the landscape, and but does Fisher not agree with epistasis as a phenomena? Or why does Fisher? That was the key thing. What I was looking for. Why does Fisher say that landscapes are going to be single peaked? Akshat.
Yeah. So, according to Fisher, Wright's understanding of landscapes is incorrect. So Fisher says that if you consider landscapes in the higher dimensional structures as they really are, epistasis won't make the landscape rugged and it will be single peaked.
Yes. Well, it would make it rugged, but it won't be multi-peaked. So as we have seen that if you have a genome of length L, and now this genome basically now if I have a genome of length L and in the sequence space there is one particular sequence, right? So that in our representation of fitness landscapes is just going to be represented by a single node, and the height of the node is going to be the fitness corresponding to that particular sequence. Now if this fitness is the maximum fitness on the landscape, then what needs to happen is essentially that this needs to be greater than all the 3L neighbors that this particular sequence has. And what Fisher is arguing is that if you have a sequence of length 2, then basically what needs to happen is that this sequence needs to be a maxima for only with needs to have a greater fitness than the 6 neighbors that it has. If it has a length 3, then it needs to have a fitness greater than the 9 neighbors it has, and so on and so forth. Now Fisher says that epistasis is true as a phenomena and it's pervasive also. And that is going to mean that there is a lot of zigzag happening in the structures of the landscape. But because the dimensionality of the structures is so large, because landscapes are such complex structures and have such high dimensionality, in order for a single point to be a maxima, it would need to be of a greater fitness than millions of its neighbors, in this case it being 3L, where L is the genome's length. It would need to be a maxima with respect to all of these points. And he says that as the genome length increases, the possibility that among the 3L neighbors, there is not even one which has greater fitness than me as that point, uh, is actually inconceivable. And hence, despite being epistasis, because of the high dimensionality of these landscapes, uh, Fisher argues that landscapes actually are going to be single peaked, because no matter where you are on landscape, there will be there will be at least one such path which leads you to higher fitness, and from there on, you can keep moving to greater heights. So he says the fact that recurrently you are going to be trapped at a point which is a local peak is not conceivable because of the high dimensionality of these plots. Does that make sense? You may not agree with it, but at least do you understand geometrically Fisher's argument? Yes, no?
I do understand, sir, but then according to him, the zigzag should also not be present, right?
Well, no, zigzag, when we say, okay, let me put it another way. How many of you know what is a saddle point? All right, many of you. Okay. How many of you know what is a saddle? Okay, so saddle is basically, what is kept on the back of the horse. Let me just, it's just easier to. Let me just share my screen. All right. So let's look at this image of a saddle, right? Now, what's happening is that we'll focus on this particular point here, which is the lowest in this particular direction, right? So as far as this direction is concerned, this point is the minima. But if you look at the same point, but we are actually looking in this direction now, this particular point is the maxima now. And that's exactly Fisher's argument. Fisher is saying that if I'm looking at a structure of a landscape and I'm looking at this particular point, because of epistasis, this point might be a maxima. But there will exist some direction in which there is some neighbor which has a higher fitness than this. So if I'm looking at the structure in this particular dimension, I see this zigzag because it goes up and then it goes down. So there is this zigzag. But if I'm looking at in this direction, the zigzag is in the opposite direction. This is a minima now. So Fisher is saying that you have uh, these variations because of high dimensionality, and the likelihood that a particular point in this zigzag arrangement of points, a particular point is a maxima in all dimensions associated with a landscape is diminishingly small as the number of dimensions are increasing, and hence fitness landscapes are likely to not have multiple peaks. That's Fisher's argument based on which he's saying that landscapes should really be single peaked. Yes, does that make sense?
Yeah.
Yeah. Do you agree?
Not completely,
Yeah.
but yeah, his argument seems sound.
So I think no one agreed with. Oh, of course, I don't think anybody wrote about Fisher in this particular context that I just talked about. Uh, but all of you tended to agree with Wright. And what I find fascinating is that remember, this was Fisher. Wright proposed the structure of a landscape in 1932, and I think it was a giant conceptual leap. We are still struggling to come up with empirical data-based structures of landscapes. As we saw in the two papers that we read, the structures of landscapes are completely different when I'm looking at a sequence space within a gene and when I'm looking at sequence space between different genes. So the structures are very different from each other. So we're still struggling in some sense regarding getting empirical data about structures of landscapes. So in 1932, when Wright proposed it, this was a giant conceptual leap. But I think what I find extremely fascinating is Fisher's this argument of dimensionality, because Fisher says the problem with Wright's visualization is that he's visualization. Remember that cartoon that we looked at of peaks and troughs from Fisher's 1932 paper, where he's saying this is what landscapes look like. And he says that the problem with Fisher's argument is, problem with Wright's argument is that he's asking us to visualize landscapes in 3D. And if you're going to look these structures at in 3 dimensions, we are going to see peaks and troughs, because that's how we are going to look at it. But he says that a real landscape has so many dimensions, it has so many ways to escape from a particular point, that you can't visualize it in 3D. It's an impossibility to do it. And Wright is limiting our visualization because he is asking us to look at these structures in 3 dimensions. In reality, the dimensionality associated with this is equal to the number of loci that are on genome. And at least at that time, in terms of loci, they were thinking of how many genes. So Fisher's argument is that the dimensionality associated with a landscape is at least the order of few thousands, because that's what the number of genes were thought to be, or at least for simple organisms. So he said the dimensionality associated with this is in the thousands. But we are being asked to look at this structure in a 3-dimensional space, where one of the dimensions is actually fitness. So the entire sequence space is being asked to be represented in 2 dimensions, which is just not conceivable. And hence, Wright's the structure itself is not wrong, but his interpretation, because he's reduced it to 3 dimensions, is wrong, and hence landscapes are going to be single peaked. That is Fisher's contention. All right. Show of hands if anybody is convinced by Fisher's argument. No one. Well, just show of hands if you still think Wright was right. Others are asleep, not interested. All right.
So Fisher raises a valid point that visualizing it in 3 dimensions is incorrect, and the argument of high dimensionality will lead to different interpretations. But he takes one problem, says that it is wrong, but then introduces another problem. So how are we supposed to visualize the higher dimensional structure he is talking about?
Don't know. I really don't know. So I mean, how would you check this? And that's an interesting point. How would you check this? And I don't think anybody has even thought of doing this up until last 1 or 2 years. So I've seen a few papers. See, what is going to happen now? Epistatic relationships are something that are studied in a very structured and a limited way. All of you know what is the gal regulon, right? I'm sorry, not gal regulon, sorry. All of you know the lac operon regulation, right? All of us, I mean, that's a classical model of regulation that's studied in any introductory class. Now in lactose regulation, so essentially what you're saying is that uh, is there anybody who doesn't know the lac operon? All right, great. So now think of a mutation where LAC-Z, which is the enzyme which hydrolyzes lactose, becomes non-functional, right? This mutation. So obviously, when I'm growing this bacteria in the presence of lactose, this mutation is a deleterious mutation. But if the same mutation happened in an organism where there was no LAC-Y was already non-functional, and now you acquire another mutation which makes LAC-Z non-functional, this new mutation that has made LAC-Z non-functional is actually neutral now, because you already don't have a LAC-Y to begin with. So you didn't even have the to transport lactose inside the cell. So the question of metabolizing lactose doesn't arise, and hence the same mutation now becomes neutral in nature. Suppose the same mutation, suppose this mutation now happened such that actually LAC-Z's expression completely changes to 0, right? In the earlier case, when the mutation happened, you were making a non-functional version of LAC-Z, right? But now a mutation happens such that after the stop codon, you immediately have a stop codon. So there is no protein synthesis of LAC-Z. Now, in this particular context, you are not making that protein at all. So what you are doing is essentially saving on the protein synthesis cost associated with making a LAC-Z molecule. And protein synthesis is an overwhelming majority of the energy budgeting of the cell. If we look at where the energy of the cell goes, things like DNA, RNA, lipids, etc., it's actually really minimal amounts of energy that's spent in making all these things. An overwhelming majority of a cell's energy resources are spent in making proteins. So now if you have this mutation where in LAC-Z you have immediately after the stop codon a stop codon, you're no longer making the protein. And if you are growing in a substrate which doesn't have lactose, then you are actually, compared to an individual which is making LAC-Z all the time, this mutation is now favorable because you are not making unnecessary protein. So in that case, picking up a mutation which stops synthesis of LAC-Z becomes a beneficial mutation. So all of these things are sort of dependent on what is the excuse me. All of these things are actually dependent on what is the precise environment in which I'm looking at this fitness. Because suppose I was looking at this fitness in glucose, then it doesn't even matter, because repression is going to make sure that lac operon is in the off state. It's dependent on the environment in I'm doing these experiments. It's dependent on what is present at other loci, depending on whether LAC-Y is present or absent. That's epistasis. So when you are studying epistasis, what's usually done is just you look at a particular system and study epistasis within it. Like the example of beta lactamase that we studied earlier in the course. If you have mutations and the effect of a particular mutation is contingent on what is present at other places in the sequence of the gene, that's epistasis. So epistasis has always been studied in this very defined and a limited context of a particular gene or of a particular system. Now, but that's not what we need to understand Fisher's argument. To understand Fisher's argument, we need global patterns of epistasis. If I characterize epistasis across the genome and classify interactions as positive epistasis, negative epistasis, sign epistasis, no epistasis, what are going to be the relative proportions of these interactions in each of these four windows? Then within the four windows, suppose I'm talking of positive epistasis. Now, not all positive epistasis cases are going to be of the same intensity. They are going to have a different intensity about themselves. So there will be a distribution in terms of the magnitude of positive epistasis that you see in different cases. So in order to get these global patterns of epistasis, we need a measure to be able to understand how are these epistatic interactions spread in these four broad classifications: positive, negative, no, and sign epistasis. And then within each, what are the patterns, what are the distributions of epistasis within these windows? That's not an easy thing to do. And up until last year, I had not even seen anything being done in order to get these global patterns of evolution. There have been at least one or two papers which have attempted this in the last 1 year. So to my mind, at least, to test Fisher's idea, you need an understanding of these global patterns of evolution and then be able to comment on something like this, right? That so far has been beyond our reach, at least experimentally, right? Suppose you were to, I mean, I guess you could test this via a simulation, that you take a landscape, you take a sequence of length L, and then you define fitness of a particular locus as, did we talk about NK models at all? Yes, no?
No.
Okay. Maybe next time. Maybe next time. But my question is the following: that suppose you defined a fitness landscape randomly. Okay, you made up some rules that fitness from this particular locus is going to depend on only on this locus and not on anything else. So fitness contribution is independent of everything else on the chromosome. But if you go to the neighboring locus, now fitness landscape of this locus is actually going to depend on what was present on this. So you have fitness contributions from each of the n loci on the DNA, and you assign these fitness contributions from each loci according to the arbitrary rules that you define, right? That some loci will have no epistasis, so their contribution is just going to be dependent on what is present on themselves. Some loci will be dependent on what is present elsewhere on the chromosome, so fitness contribution of this loci will actually depend on what's present here. So these two combined will ensure what is the fitness contribution of this loci. Another loci might actually be dependent on two other loci, so this triplet will decide what is the fitness contribution of this loci. And then some of these epistatic interactions are going to be positive epistasis, negative, and so on and so forth. Suppose you made up these rules, right, and then created a fitness landscape. Then the question is that as the dimensionality, Fisher's argument is that as dimensionality increases, the chances that you have a fitness peak, you have multiple fitness peaks in this structure, diminish, right? That's his argument. And it's a question that has remained outside the bounds of empirically testing it so far. But I don't know how you guys feel about it. Does anybody feel convinced by Fisher's argument? No one. All right.
Slightly, if we add the things we learned about neutral mutations into the argument,
I'm sorry, say that again.
So when they had this debate, there was no consideration for neutral mutations, right?
No.
So if we add consider neutral mutations can also be present, then there is more credibility to Fisher's argument.
Yeah, agreed. Yeah. Yeah, no. I mean, I empirically being able to test the idea is one thing. But I at a very academic level, I am actually blown away by the argument that Fisher is proposing. I think what Wright proposes is something very elegant and geometrically very beautiful. But I think what Fisher is saying, he's using that to counter Wright's own argument, and I think it's actually really cool what Fisher is proposing. I mean, I don't know if it's right or not, and as we will see in this section on experimental evolution, that fitness landscapes are not so easy to define. Uh, but that's we'll come to that in due time. All right. Anyway, that was what I was looking for in Fisher's. That was the crux of the debate between Fisher and Wright. That it wasn't about Fisher's fundamental theorem. It wasn't about the fact that Fisher said that there is no epistasis. Fisher said there's definitely epistasis and it's pervasive in nature. But despite that, Fisher is saying that it's going to be single peaked just because of the dimensionality issue. So thinking of the saddle example again, what Fisher is saying is that in order for one point to be a maxima, in every dimension, it would have to be maxima. So in a saddle, we saw that in one dimension, the point is maxima, in the other dimension, it's a minima. But if there are thousands of dimensions, Fisher's argument is that for a point to be a maxima, that point needs to be a maxima in every single one of these thousands of dimensions. And that, he says, is really unlikely, just because there are so many dimensions, and statistically speaking, there would always be a dimension where you can escape via a mutation, which is beneficial in nature, and go to high fitness. So hence the probability that a point is a fitness peak decreases as dimensionality increases. In order to test this, you could design a simulation where you have a dimensionality. You have L as the number of loci, and you have a distribution of epistasis, fixed distribution of epistasis. That 40% of the cases are negative epistasis, 30% are positive epistasis, and so on and so forth. Right? And within them, you have these distributions of epistasis. Now you are looking at, suppose you were to, so you define these epistatic interactions. So in effect, you have the entire fitness landscape with you. Now what you're saying is, within this structure, if I were to pick a point at random in the sequence space, what is the probability that this point is actually the fitness peak? And you have this as a function of L. So on the x-axis, you have L, and on the y-axis, you have probability that a randomly chosen point is a peak. And Fisher's argument is that as L increases, the probability that a randomly chosen point is a peak decreases, and hence in the extreme case where L is actually thousands, or if you're talking nucleotides, millions, the probability that a point is a fitness peak is essentially zero. That's his argument. And that's what I was looking at in this submission. Okay, if there are any comments, we can take them. Otherwise, we can start with our discussion today.
So, what exactly is Fisher-Wright model? Is it something related to this simulation and testing only?
No. So you remember Moran process?
Yes.
Right. So in Moran process, what you are doing is essentially you are tracking the evolutionary trajectory of a population. And the way to do that in a Moran process is that in every time step, one individual dies and one individual is born, and you update the structure of the population and you keep moving forward. Now, it's easy to implement and so on and so forth, right? But the problem with that is that computationally it becomes really hard to trace. As the size of the population increases, it becomes increasingly difficult to trace it for a very long period of time, right? Does that make sense?
Yes.
Yeah. So Wright Fisher model is is a is a is a model to study exactly that, evolutionary change in a population, except for the fact that it allows you to study evolution of a population where each time point is actually one generation. So in Moran process, one time step is not one generation, because in one generation, if you have n individuals, in one generation, these n individuals are going to be replaced by the progeny n individuals, right? So you would need n steps of the Moran process uh, to go from one generation to another, right? So in that sense, a single step of Moran process is one nth of a generation, because when you take n steps, you have one generation. But in Wright Fisher model, the single step itself is one generation. Does that make sense?
Yes, sir.
Right? The other difference is that in a Moran process, if this is your starting population, if you simulate it using a Moran process, you are going to get an evolutionary trajectory of the population. Of course, if you do the same simulation again, you will get another evolutionary trajectory, and so on and so forth, right? If you do this millions of times, right, you will get a probability distribution that after one generation, this is what the probability distribution of the population makeup is going to look like. Does that make sense?
Probability distribution of exactly what?
Probability is, suppose there are, so let's go to the marbles in a jar game. Suppose you started with 5 blue marbles and 5 red marbles. After one generation, you have a probability distribution of how many blue and how many red marbles are going to be there in the jar, right?
Okay.
Right. So in a Moran simulation, you don't get the probability distribution. You get outcomes of one of the trajectories, right? And if you simulate this many times, then you can derive the probability distribution out of that. In a Wright Fisher model, you go from t equal to 0 to t equal to 1 generation in a single time step, and what you get is the probability distribution of the population structure at one generation and then subsequently following generations. Does that make sense?
Okay, so like it is similar to Moran process only.
It is similar to Moran process. It's a probabilistic model of tracing populations where each time step is one generation. Yeah.
Okay.
It doesn't have anything to do with the debate that we discussed.
Yes.
All right. Any other comments before we move ahead?
Just one thing.
Yeah.
आपने मेंशन किया कि फिशर का argument was probability of any random sequence point being the peak decreases as the sequence length increases.
Yes.
But ignoring the debate, that would be expected because the number of points in the sequence space increases exponentially if you increase it. So the drop in probability is natural to expect, right?
Yeah. Yeah. So can you normalize for that effect?
Can be.
I don't know, but the point is that if you have two peaks in a genome sequence of, or if you have 10 peaks in 10 million, right? I mean, that's not, see, it's a qualitative argument in some sense. Fisher is for Wright, the argument is, for Wright, evolution is explaining the following. For Wright, evolution explaining evolution is answering the following question: how does a population which is stuck in maxima, in a local maxima, move out of there into its global into the next higher peak, or any other maximum, right? That is the problem for Wright as far as explaining evolution is concerned. Now what he anticipates is that this is happening constantly, right? A population is constantly getting stuck in a local maxima, again moving out, again getting out. So he says the movement dictated by selection is actually a trivial problem, and that's not the crux of the problem as far as explaining evolution is concerned. It's about moving from a local maxima to another local maxima. That is what is needed. For Fisher, that's not the case at all. He's the other extreme, who says that there is only one peak, so natural selection actually explains everything. You don't have to invoke concepts like drift at all. Natural selection would do anything. Now what you are saying is that there is a middle ground there, that that there may be 5 fitness peaks in the landscape, right? So I would still say that's not right. That's closer to Fisher's argument rather than Wright's argument, because even if a population is stuck, for Wright, the structure is so rugged that you are just constantly moving from one fitness peak to another fitness peak, and that's not going to happen in a scenario where there are only 5 fitness peaks or something like that. So that's the argument. I mean, there is some middle ground, obviously, but does that answer your question?
Yeah, makes sense.
Yeah, I don't know. Pata nahi. We'll see. Okay. I mean, in this experimental section, the one study that we will look at quite carefully is an evolutionary experiment. How many of you have heard of this long-term evolutionary experiment with E. coli? It's called LTEE. All right. So we'll spend at least 2 classes on this experiment, which was started in 1987, where somebody started evolving E. coli in one particular environment, in low glucose environment, right? And the experiment is still going on. And obviously it's been 30 something years, and the number of generations that they have processed is now close to, I think, 75,000. So this is a neat study for us in order to study movement on landscapes, because you would imagine the environment is still the same. It's been going on for tens of thousands of generations. So if there was a fitness peak, or if populations were getting stuck in a fitness peak, then they would have done that, and this then becomes a great model to study that question. And we'll see some of the complications that you run into experimentally when you're trying to theorize these concepts via the structures as envisioned by both Wright and Fisher. So the dynamics of evolution is much more complex than both what Wright and Fisher envisioned. And we'll see. And what's interesting about this experiment is, when this person started this experiment in 87, he didn't start one line. So he didn't have one population which was he doesn't have one population which has evolved for 75,000. He has 12 replicates of it. So the starting population in each of the 12 replicate populations was the same. It was the same ancestral E. coli which has been propagated. But since that day in 87, these 12 populations have not seen each other. They are independently evolving in identical environments. So you have that question of repeatability also, that you can ask that are these 12 populations getting stuck in the same fitness peak, or are they getting stuck in different fitness peaks, or have they all reached the same point on the fitness landscape as far as peak fitness is concerned? Was Fisher right? And 75,000 generations in the same environment is a long enough time that this gives you a lot of insights into how actually evolution would proceed. And of course, this has also been done in a time. If this experiment was done 30 years before, then we wouldn't have had the chance to sequence stuff. But this has been done in a time where sequencing is now not very expensive. So you can really extract a lot of information from this. Does that make sense?
Yeah.
Yeah,
And
yes,
they
go ahead.
are they also accounting for genetic drift?
See, you should look at it as following, that when you do an evolution experiment, right, and we'll discuss, there are 3 standard ways of doing an evolution experiment. We'll discuss them just in a bit. But when you do an evolution experiment, you are not, what you are seeing. So suppose I start with a sequence. I evolved this population in a particular environment for 75,000 generations. And at the end of this 75,000 generation period, the environment remained constant throughout, right? I get this evolved population. So I get this evolved population sequenced, and I get this ancestral population sequenced, and I'm studying the genetic changes that took place in this intervening 75,000 generations. Now, what these changes I'm seeing are a sum total of changes that took place. Some of these changes would have taken place because of selection. Some of these changes would have taken place because of drift. Some of these changes could have also meant that actually deleterious mutations reached fixation in this population. And one of the mechanisms of doing that, of deleterious mutations getting fixed, is called hitchhiking. That suppose I have a deleterious mutation on the genome, but I pick up this fantastic beneficial mutation, which just moves me to the front edge of the traveling wave model that we did. That I was actually doing very badly in the population, but mutations are a chance event, and I just happened to chance upon this great beneficial mutation, which placed me right at the front of the leading edge of the traveling wave, and hence I reached fixation. So even though I was carrying this deleterious mutation, because of this new beneficial mutation, the deleterious mutation also got fixed. So I guess what I'm trying to say is that at the end of the experiment, the genetic changes that you see are a sum total of changes that have taken place because of all factors that influence evolution. Now it's up to you to differentiate that which changes were selective in nature, which changes were because of drift, and which were perhaps deleterious and just got fixed because of because of very rare chance events. Does that make sense?
Yeah.
All right. Any other questions, comments? Okay. All right. So in experimental evolution, there are really 3 factors. One is the choice of organism that you have, right? Then the environment that you are going to do this evolution in. And when we say environment, we speak of both the biotic and the abiotic factors. The abiotic factors can be the physical and the chemical parameters. That are you doing this experiment in in nutrient deprived conditions, nutrient rich conditions, and so on and so forth. Other abiotic factors could be, you are doing this at pH 5, you're doing this in presence of antibiotic, and so on and so forth. At what temperature are you doing this? So all of that comprises abiotic factors. Another important factor, which is largely not looked at in experimental evaluation, is biotic components. That when you do an evolution experiment, you're actually doing this in lab. I would say more than 99% of all evolution experiments in lab are done with one species. And the critics of experimental evolution as it stands today will say that that's not representative of any sort of ecological evolution that takes place out in the nature, because any niche you take, you just do not have one species, but you have multiple species coexisting and interacting with each other. So I mean, there are pros and cons, but this really allows you to focus and study what happens, but you're not representing an ecological niche and so on and so forth. So there is this tension between evolution and ecology, because in an ecological setting, you have no control over the experiment. You can't do molecular stuff. You really don't even understand the environmental pressures that are out there in the population. So the evolutionary people will say that, oh, you have no control over the population. There is no constancy, so we don't know what you're looking at. But on the other hand, the evolution people are critiqued by saying that, you are just doing something in a constant environment for 75,000 generations, single species. That does not happen anywhere. So while it tells you about what happened in this particular environment, in this particular experiment, that doesn't tell you anything about how evolution proceeds generally in any ecological niche out there. So there's this tension between these two fields. And the third bit is sort of the interesting one, which is mode of propagation, because an evolution experiment by design is an experiment which propagates for many generations, right? So how many of you have grown a bacterial culture before? Okay, that's it. Anirudh, how do you do that?
Well, I don't remember the exact specifics. I was in undergrad, but the way I would do is you have.
Wasn't that long ago?
No, I worked with cell cultures. The normal human cell culture is more.
Okay, sure.
We used LB media, and so it would be in a flask.
Yeah.
And we put one drop of. We'd first autoclave it, and then we put one drop of bacteria into it, and then we just we have a shaker. We cover it and we put it in the shaker and this.
Cool. Right. I mean, a eukaryotic line is not going to be any different. You're looking at what you would call an expansion in that case, right? So there's really no difference there. So what do you mean by one drop of bacteria?
Where you measure. So you measure the concentration that you want to put in that bacteria. You take a micropipette and you figure out the concentration, and you take that much amount and place it in. It's not one drop exactly, but it's based on how much concentration you want in.
Right. So let's make up some numbers here. So in your flask, how much LB did you have? Just order of magnitude.
In mLs, I think. Like
yeah.
I don't remember.
Make up something.
Okay, 50 ml.
50 ml is great. Huh? Okay. How much did you pipette in?
Microliters, some amount of microliters.
Make up something.
2 microliters.
2 microliters. Fine. So you took 2 microliters of, I'm assuming, saturated cultures, right? And you allowed them to grow in 50 milliliters, right? So obviously, let's call the number of bacteria that were present in the 2 microliters as equal to N naught.
Okay.
Right? And when you come the following day, your 50 ml flask is going to be saturated. And let's call the number of individuals in that flask as NF, N final. Right?
Yeah.
What would be the factor? What is NF by N naught?
Would it be the ratios of the volume?
Yeah, exactly. Right. So it's just going to be 50,000 divided by 2, 25,000. Right?
Yeah.
Does everybody see that? So you have 50 mils of culture, and you started that culture with 2 microliter of a saturated culture from a previous tube. And when the 50 ml flask gets saturated, the number of bacteria has increased by a factor of 25,000, because you have 50,000 microliters and you initially only added 2 microliters bit of saturated cultures. Does that make sense? That's pretty straightforward. Yeah. So how many generations is this? That's the question that we are interested in, because in evolution, it's not really the time of an experiment. The key variable in evolution is not the time for which you did the experiment, but the number of generations that you propagated. For instance, if you're doing experiments with human populations, right, it makes absolutely no sense to say that I did a evolution experiment with a human population for 10 years, because I mean, you reach sexual maturity in 10, 15 years. So I mean, a generation is 15 years. So it makes no sense to say that. So the relevant time parameter that represents an evolution experiment is how many generations did you process in the experiment. So in the experiment that we are discussing, this 25,000 fold expansion in the number of cells that took place, how many generations is that? How would you calculate that?
So maybe 2 raised to the power n equal to 25,000.
Say that again, Anthony.
2 raised to the power n equal to
Yeah.
25,000. So n is the number of generation.
Correct. Right. So essentially, you do it as exactly what Anjani said, that if you're starting with N naught, in one generation you will get 2 times N naught cells. In 2 generations you will get 4 times N naught cells, or 2 to the power 2 N naught cells. In 3 generations you will get 2 to the power 3 N naught cells, and so on and so forth. So you are interested in an n such that 2 to the power n, let me just write this down. I don't know how many generations that is. So if you're starting with n naught, in one generation you have 2 to the top 2 times n naught. In 2 generations you have 4 times n naught. 3 generations 8 times n naught, and so on and so forth. And you are interested in n such that 2 to the power n, well, n is a bad choice of variables, but let's say n generations, 2 to the power n times n naught is equal to 25,000 times n naught. Right? So this happens at saturation, and you're interested in figuring out this value of n. So n is just going to be equal to log of 25,000. So that's 15 generations. So that what you have done in that experiment is you have processed 15 of the order of 15 generations in a single day. Now that's perhaps possible for an organism like E. coli, wherein 24 hours the culture reaches saturation, but that may not be possible for some of the organisms that are going slower. So, but the key here is that you process 15 generations. Now, 15 generations is really not a whole lot as far as experimental evolution is concerned. So you need to propagate this further. What would you do? Because in 15 generations, your culture has reached saturation and there is no more nutrients left. So what would you do?
We'll take a small sample and repeat it.
You take a small sample and
Put it in another flask.
exactly, right. So that is the most common way an evolution experiment is done. That after it has reached saturation, you will take a small volumes, let's say again 2 microliters, and you will put that 2 microliter in a fresh flask which has 50 mLs of uh, autoclaved of sterile media, and you repeat this whole process again. So every day you are processing 15 or so generations, right? So if you do this in a week, you will do 100 generations. Does that make sense to everybody?
Yeah.
What are some of the issues that you can foresee with an experiment like that? And I mean, logistically it's pretty straightforward, but are there any technical scientific issues that you need to be careful, that you need to take into consideration when you're doing something like this?
There is added amount of selection based on chance. I mean, we are only taking a small sample from this population, and this might not represent the whole population, right?
Yeah, exactly. Right. So that's one. And again, we are not saying this to discount this evolutionary strategy. This is actually the most widely used experimental strategy by any lab in the world. But for instance, so we have 15 generations. Maybe let's imagine that a beneficial. Okay, let me put it another way. We have 15 generations. How many? In which generation are the maximum number of cell divisions going to take place? Uh,
14.
yeah, and to make things simple, let's say N naught was 1. You added 1 bacteria to the culture. So how many divisions took place in the first generation? Only 1. In the second generation, you will have 2 divisions take place. Third generation, you have 4 divisions take place, and so on and so forth. So the maximum number of cell divisions are going to take place in the last generation in your culture flask. What does that mean? What are going to be the implications of that? Want you guys to tell me.
Can you repeat that, please?
Yeah. So you're starting, your N naught is 1, right? So in the first generation, 1 division take place. In the second generation, so this 1 became 2. That's after 1 generation. In the second generation, both these divide, so 2 divisions take place. So now you have 4, and so on and so forth. So in the 15 generations that you see happen in this 24-hour period, in which generation are the maximum number of cell divisions going to take place? Is the question clear, Anil?
Yeah, the previous to the last generation, right?
Right. So that's fine. So this last generation where the population was NF divided by 2, that's going to be the size of the population, NF divided by 2, and you have NF by 2 divisions taking place such that the population reaches a size NF. What are the implications of this as far as an evolutionary trajectory of this population is concerned? Anybody?
Is it that the population will be briefly in the saturated growth period range?
Yeah, we'll come to that, but that's not what I mean. What I'm trying to get at is that you have a maximum number of divisions taking place in the last generation, right? And remember, this is exponential, so this is heavily biased. If you had a population size of 10 million, 5 million of those divisions took place in the last generation. Now the chance of a beneficial mutation, if you forget deleterious mutations for a bit, they're going to be weeded out anyway, so let's forget about them. If you're thinking of beneficial mutations, the we're not, well, it's only beneficial mutations which are going to alter the course of evolution, so we are only talking about them. The chance that a beneficial mutation arises is proportional to, let me say that again, let mu be is mu is the mutation rate, and if mu is the total mutation rate, and if 10% of those mutations are beneficial mutations, then 0.1 times mu is the rate of mutation for beneficial mutations. That's mu b. In every generation there is a n mu b chance. The number of beneficial mutations arising is n mu b, where n is the number of divisions taking place in that generation. Does that make sense?
Yeah.
Yes.
So given that, what is the most likely time that a beneficial mutation is arising in this culture that we are talking about?
The generation with the highest division.
Exactly, which is the last generation, right? Now if the greatest chance of a beneficial mutation arising is actually the last generation, what is going to be the frequency of that mutation in the saturated population out of which you are going to take a 2 microliter sample? What is the frequency of that beneficial mutant?
Very low.
1 upon n,
Yeah.
right? 1 upon nf. And hence that beneficial mutation becomes extremely susceptible to drift. And drift in this case being that of the total pool of NF individuals, you are only taking you are only taking NF divided by 25,000 of the individuals. That's what you are taking to the next culture. And now you are relying on the fact that one beneficial individual is going to get picked from this population and get transferred to the next one where it can establish itself. Remember that first few phases are going to be establishment, where it can establish itself and then sort of work towards fixation. Right, again, not we are saying this not to discount this experimental technique. Everyone uses that, bar no exception, but we need to be mindful that this is what's happening in this culture. Does that make sense?
Yeah.
Questions, comments with this? Okay. Now let me move forward a little bit. So if this was N0, this is my N0, and we know the growth trajectory of a bacterium in a population looks like this. Does everybody agree with that? There is a lag phase, then there is a phase which is called log phase, exponential growth, and then as the nutrients begin to get extinguished or get exhausted, you have this stationary phase, and then there might be a death phase also after this, right? But this is a typical trajectory of a growth curve of a bacteria in a batch culture in a flask or a test tube. Everybody agrees with that?
Yes, sir.
One thing I should add before we discuss this is that I make it sound as if cell division operates like a clockwork. That there's we all read in textbooks that E. coli divides in 20 minutes, right? Has everybody heard that in LB media 37 degrees E. coli divides in 20 minutes, right? So does that mean that every single cell is going to divide at 20 minutes like clockwork? Suppose my N0 was 1. In 20 minutes, I'm going to have 2 individuals, and then nothing happens. They grow in size for the next 20 minutes, and at 40 minutes, if I look at 40 minus delta, I'm looking at 2 individuals, and if I'm looking at 40 plus delta time, I'm looking at 4 individuals. Does it operate like that?
No, I think it's considered like a random variable around the mean 20.
Yeah. Rachita?
Same, sir. It wouldn't happen like that.
Yeah, it wouldn't happen like that. In fact, one of the somewhat surprising things is that when we are looking at eukaryotic cell cycle, a lot of that is understood, right? We know that there is G phase, there is S phase, and so on and so forth, right? So we understand the time that a cell spends in these phases, and then cell divisions happen when you do a full round. But a lot of cell cycle associated with bacteria is actually not known, and the checkpoints associated with and the dynamics of these processes are not known. But what we do know is that cell division times are not are obvious excuse me, are not like this clockwork feature. There is a distribution obviously of these division times, and these distributions are, again, work done in the last 5 to 10 years has found out that these distributions are represented by what are called log Fréchet distributions. Okay, essentially these are normal distributions, but they have an elongated tail. So there are some cells. So on the x-axis is the division time, how long does a cell take to divide, and y-axis is the frequency of the number of cells. And this is the mean, which is 20 minutes. So it's not as if everybody is dividing at 20 minutes. You have a distribution of division times, but there is a long tail of this distribution. Like there are some cells which take a really long time to divide, and there are proposed mechanisms as to why this could be so, right? So there is a distribution. So when we paint this picture that the maximum number of divisions are happening in the last generation, I make it sound as if there is a burst where you have cell divisions taking place every 20 or however many minutes, depending on the media, taking place. That's not really true. But what's important is to understand the argument qualitatively rather than quantitatively. So while it's still true that most of the divisions are going to take place in the last phase of the growth, it's not strictly true that it's going to be that one. The generation time is not like clockwork for the entire population. A bacterial population is all asynchronous, where cells are in different phases of growth. Isogenic cells are in different phases of growth. if you were to take a snapshot of the population at any given instant. Okay, so this is a typical growth phase of a bacterium, right? Now let's say one of us is doing an experiment where you add N0 cells to the media, you let the culture grow, and as soon as the population reaches the size which is represented by this point A here, you subculture. You take N0, so let's say this number is Na. From this Na, you take N0 cells and you start it again. So one of us is doing this evolution experiment, that the population reaches here, you again subculture, reaches here, you again subculture, and so on and so forth. Another one of us is letting the population increase to B, so NB, it reaches here, and then you subculture and you keep and you sort of operate in this loop. And another one of us is letting the population reach C, which is the maximum carrying capacity of this environment, this test tube or flask. And once it reaches C, that's when so the cells spend some time in the stationary phase, and then you take N0 cells out of that, and you let the experiment continue. And let's say that you do this, you repeat this process for 1,000 generations, right? I mean, because the amount of expansion allowed in one cycle is different. In the A point, maybe there are only 3 generations before you subculture, and in the C point, maybe there were 10 generations before you subculture. So the number of generations is different in each case. But let's imagine that you did this experiment for an equal number of generations, each of these 3 experiments, and then you do a growth curve. So the outcome from this experiment, you will get one genotype from the outcome of this experiment, you will get one genotype from the outcome of this experiment, and this one, and then you do growth curves of these evolved lines which have been evolved at point A, B, and C. What would your expectation be regarding the growth curves that you get from these 3 points, A, B, and C? Before you think about that, is the question clear?
Yes.
Others?
No, can you repeat the last part, please?
Yeah. So let's say, so the experiment is clear, right, Anirudh? Like you evolve it and let it grow to A, B, or C, and then subculture again, and you do that for 1,000 generations in each of the 3 cases.
Yeah.
Right. So let's say you were doing the experiment where you evolved it till A. Akshat was doing it where you evolved it till point B, and Prachita was doing it where she evolved it for till point C. Right. Now at the end of this experiment, each one of you has an evolved line, right? Your is evolved line A, Akshat's is B, and Prachita's is C, right? Now you add equal number of cells, N0 cells. Okay, so you add equal number of cells, N0, in a 50 milliliter flask. You add your evolved line, and the other two add an equal number of cells, not in the same flask but identical flasks of their evolved lines, and you characterize the growth phase. You characterize the 3 phases of growth of each of these 3 evolved lines. My question is, one, what differences do you expect of these evolved lines as compared to the ancestor? And second, what differences, if any, do you expect to have evolved in these 3 lines that you have between the 3 of you. Does the question make sense?
Yeah.
All right. Anybody, any questions with what I'm asking? Forget the answer, but any question with what I'm what the experiment is and what are we looking at? Okay, thoughts. Tell me what you think.
Experiment C will show the highest deviation from the ancestor.
Okay, maybe I don't want to get into it. What I would like you guys to do is sketch this. Sketch the growth curves of A, B, and C and the ancestor on a piece of paper, and send that picture. You don't need to make anything neatly, just as long as it's legible, just pen and paper would do. Just take that image and send it to me before Friday's class. We will start the class with this discussion, and anybody who doesn't send me will be asked to leave the class, send it to me, and then come back again. Right? But I'm interested in what you think. Okay, let me make one more point, and then perhaps we can stop for today. When you do so, this type of an experiment, so if we look at these notes here, I said there are 3 typical ways of doing an evolution experiment, right? What we are discussing is the second one, and this is called serial transfer. So add n naught number of individuals, allow for growth, population increases to n, nutrients are exhausted and no more growth, and then you transfer n naught number of individuals to a fresh tube, and so on and so forth. You keep going on and on like this. So by using the same logic, the expansion that we discussed, so this expansion from n naught to n final was 25,000 fold in the scenario that we discussed. A typical number used in an experimental setup when you are using this particular technique is 100-fold dilution and then 100-fold expansion, which comes to about 6.7 generations in each transfer. And now that each transfer could happen every 12 hours in some of the experiments, for instance, that we've done, it could be several days if you're working with an organism which grows slower as compared to E. coli, which most organisms do. Right? One limitation of this, so again, we are discussing all these, maybe all these limitations, some of them which might seem fatally flawed to you, but please do remember this is used in more than 99% of all evolution experiments. And Friday, when we discuss the other 2 techniques, it will become clear why the others have limitations about them. And this one does also, but logistically it's just a lot easier to handle. Essentially, you just have a culture growing, you come and just subculture, and that's it. You're done for the day. Right? One of the other limitations of this particular experimental technique is that the environment doesn't stay constant. And what we mean by that is that when I say that those guys have evolved E. coli in low glucose environment for 75,000 generations. But the point is that when I do the subculture, suppose my glucose concentration is 5 grams per liter in the media that I have made. Now, at the start of this experiment, when I have N naught cells, the amount of glucose that's present is 5 grams per liter. I mean, the concentration of glucose present is 5 grams. And the same goes for other nutrients as well. but in this case, glucose is the nutrient which is limiting growth. If you were to increase anything else, growth wouldn't increase, because growth is being limited by one source here, which is the amount of carbon that you've added to the media. So the problem with this approach is that the environment doesn't stay constant over this period of 24 hours that you're doing this experiment before you subculture. Obviously, nutrients are decreasing, right? And hence the selection pressure that the cell is facing changes as the propagation, as the growth takes place in that 24-hour period, right? The other thing that changes the environment is the fact that you have more cells. That means that some of the metabolic waste products are going to be secreted into the media, and that could pose a challenge or could pose an opportunity for the individuals in the population existing, right? One of the observations which has been made dozens of times is that when you evolve E. coli in high glucose environments, a curious thing happens. There is a concept called overflow metabolism. I don't want to get into the details of that, but the point is that when you grow E. coli in high glucose environments, it likes to convert that glucose into acetate, right? That's incomplete utilization of all the energy that's available in a glucose molecule, but it doesn't completely oxidize the glucose into CO2, right? Or it does that, but it does that for a very small fraction of the glucose it's using. The large fraction of the glucose that it's using, it's simply converted into acetate, and it's thrown out into the media. And while E. coli has the ability to use acetate, it doesn't use that acetate that it's throwing out because there is glucose available in the media. Now what happens then is, once all the glucose is gone, now it doesn't have any glucose to feed on, but there is this acetate that it threw out earlier. And now this E. coli starts to use that acetate. And what happens in such a scenario is that two populations emerge. You have coexistence of a mutant emerges, and this mutant is actually great at using acetate, right? It's not so it's great at using acetate, and the regulation of acetate utilization and glucose utilization is such that if you are good at glucose utilization, you are bad at acetate utilization, and if you are good at acetate utilization, you are poor at glucose utilization. So what happens in evolution experiments which are performed in high glucose is you get two different type of genotypes which are coexisting in the population. Every day when you subculture, in the first phase, these glucose specialists, what they're called, grow fast, and the acetate specialists are really struggling because they're not very good at utilizing this acetate. But when these glucose specialists are doing very well, they are also releasing acetate into the media. And when all the glucose is exhausted from the media, these glucose specialists have to switch to acetate, which they are terrible at. But these acetate specialists do very well on acetate, and they catch up. By the end of the growth phase, when all the acetate and glucose is gone, you have some fraction of glucose specialists and acetate specialists that make up the population. And then you transfer, and the same story repeats itself, right? So you have stable coexistence of two different genotypes within the same tube in the same evolution experiment. And this is one of the observations that has been seen many times over the last 20 years or so. The point is that this was only possible because environment changed during the course of that 24-hour period between one subculture and the other. So one of the things that you have to be very careful about when you're doing an experiment like this is understand the selection pressure suppose you're trying to evolve for antibiotic resistance, right? You have to understand the selection pressure that you are imposing on the organism that you are studying in any particular case, because while you may create an environment at t equal to 0 and think that this is the selection pressure under which I'm evolving this cell, but as the 24 hours progress, if that selection pressure is no longer the dominant selection pressure 4 hours after growth, then from t equal to 4 hours to t equal to 24 hours, the organisms are actually growing under a completely different selection pressure, and the beneficial mutations that you are going to select for are going to be from the context of this new selection pressure, which was in operation from t equal to 4 to t equal to 24 hours, and not the initial selection pressure that you thought you were applying from t equal to 0 to t equal to 4 hours. So this variability creates this need for you to think very carefully about any evolution experiment that you are designing so far. I mean, I've discussed this in abstraction, but we will discuss many examples of experimental evolution in the coming weeks. All right, let's stop for today. Unless there are quick comments, questions, we can take one or two of them, and then we'll continue this on Friday. Anybody? Okay, great. I will see you guys on Friday, but I do need that piece of paper sent to me before Friday, and entry into class on Friday is contingent on you having submitted that piece of paper. All right, if there are no more questions, we can stop and feel free to leave.
Thank you, sir.
Bye.
Sir, regarding the mid-sem feedback,
Yeah.
sir, around what time did you send the mail? Because I don't think I received it. So if you could.
I may not have sent it to you because in most of the things for pretty much everybody, what I did was made notes in the submissions itself. I think yours was handwritten.
Right.
Yeah. So let me just check my email and notes, and then I'll get back to you. So,
Okay. Thank you, sir.
no problem. Bye.
Bye.
Yeah, I had a question regarding the same submission we
Mhm.
had. So in
Yeah.
that, basically, you're just asking us to write. I mean, draw the whole growth, the whole the phase as we saw the S graph that we see.
Yeah.
But only after 1,000 generations, given that we take from A, B, or C.
Yes.
Okay. That's it.
So think of it like this, suppose you were doing an experiment where the culture reached saturation after 16 hours, okay? But you were subculturing every 48 hours. Okay. So essentially, you might think that and this experiment was done on low glucose environment, okay? And your premise behind this experiment may have been that I want to isolate mutants which can grow faster on low glucose. Okay. Right? Because the idea being that anybody who goes faster is going to take over the population, and hence I'm going to select for those mutations which confer faster growth on low glucose. Right? That may have been the premise, but my guess would be in an experiment done with these logistics, you will not be selecting for that because the culture reached saturation at 16 hours. For 32 hours, it was in starvation mode. So what you are actually selecting for is mutations which increase survival capacity in stationary phase. So the mutations that you're going to select for are not going to have anything to do with growth rate on low glucose, but they will have everything to do with being able to survive the longest in stationary phase.
Okay. Yeah.
So depending on when you subculture, the selection pressure changes. That is what the last point that I was making, that during the course of the time between when you first inoculate and when you subculture from that same flask, the selection pressure changes. And the point of that APC is to recognize the fact that that selection pressure, you're selecting for different things depending on when you subculture.
So
Right.
so in that case, the linear phase would be the best region.
Well, what is best?
I mean, if your goal is to
Suppose so, we our model system, the system that we work on is a very weird sugar, and we work with yeast. Okay, and it's weird in the sense that the lag phase in our culture is of the order of 2 days.
okay.
So now if I'm subculturing from deep stationary phase, then I'm again selecting for individuals which survive the stationary phase stress, right? But if I was selecting for individuals with shorter lag phase, right, when should I subculture? Suppose I wanted to isolate mutants which could halve their lag phase from 2 days to 1 day. That's the mutation I was interested in, right? When should I subculture? Suppose I
Probably
was
in the lag phase itself.
when in the lag phase? So I mean that's exactly what I want you to think. Okay, that depending on what is the exact question, the setup and the dynamics and the logistics of this experiment are going to change, and that's what I want you to think about through this exercise.
Okay, so we just have to draw 3 other graphs, each
You have
representing.
to send me 4 curves. Yeah.
Okay.
Yeah.
Okay. Thank you.